Mic check. Alright, hi everyone, welcome back, and let's get on with our lecture. Before we start, I will have a few Top Hat review questions, so let's do some review from our previous lecture. So if you have Top Hat open, and again, our join code is here. So 310274. Some time to join in. Alright, let's start the question. So the question is, can you put these in order in their hormonal axis? So we're ignoring the negative feedback for this question, but what's the order in which these are activated in this axis? So we have what? We have adrenal glands, we have the hypothalamus, we have the pituitary gland. What's the order in which they activate each other? So for this question only, we're talking about activation, no negative feedback in this question. Okay, let's see the responses. This is the most popular order, hypothalamus, pituitary, then adrenal glands, and this is correct. Again, with the hypothalamus, pituitary, or any of these HP axes, it's always hypothalamus, pituitary, and then whatever the third organ is, depending on the hormone cascade. Now that we've established, okay, in this HPA axis, it's hypothalamus, pituitary, then the adrenal glands, can you put the hormones in order? And there are some red herrings here. So you're not going to use all of them, but which fits into 1, 2, and 3 in this HPA axis? And don't worry, like you won't get a tricky question like this on the test, but it's just like, okay, do you know which fits into this axis? Alright, so what we see is the most popular matches are here, and this isn't quite right. So with this is like corticotropin releasing hormone, then we have ACTH, and then we have our glucocorticoids including cortisol. Yeah, that was a little curveball there, but glucocorticoids, remember cortisol, is part of this axis, and the other ones, choices like TRH, TSH, that's in the thyroid axis, so that would be the HPT. But in this one right here, we're talking about the HPA axis. So again, you do have that releasing hormone, the first step, but what's the end product? That determines what the first two steps are. Yeah, I know it's like, that was a little tricky, but again, it's just showing you like, okay, there's commonalities, but there's also differences as well with the different hormonal axes. Okay, so let's go back to our presentation. Yeah, maybe something, I'll try to, see, they're not showing, yeah, it only shows like 33 of you were having it, so I'll see if there's something up at Top Hat. But again, maybe it's like the format of the question, maybe it's like something with the sorting. Or it could be my computer. Nah, it's probably like something on like with the format of the question. Okay, so let's talk about endocrine, but now we're moving on to a different set of hormones. So remember that, like with our general adaptation syndrome, and this is why I bring you up this HPA axis again, we have our hypothalamus releasing CRH that binds to receptors on the pituitary gland that triggers them to release ACTH, and then ACTH targets the adrenal cortex, again, that part of the adrenal gland that's not as inner and deep as the medulla, but this is a very important part with this multiple layers. But the cells that have this ACTH receptors, they are able to, once they bind ACTH, then they produce cortisol as when they're activated. So cortisol, what's it going to do? It's going to increase blood glucose concentrations. Now cortisol is also not only just a stress hormone, and how does it do that? Well, one way that cortisol increases the concentrations of blood glucose in your body is by targeting the liver. So the liver has receptors, glucocorticoid receptors, and when cortisol binds to these glucocorticoid receptors on the cells inside the liver, these cells are called hepatocytes, but we cover them more in detail later on in the semester. But what they do is when cortisol goes to the liver, it triggers a process called gluconeogenesis. And you're like, ooh, this might be a new word. And if you like breaking down words into its parts. And if you've taken my class before, you know I love doing this. But why? Because these root words appear over and over in medical and scientific terms. So glucose sounds like glucose, so that's pretty easy. And then neo refers to something that's new or creating something. Or actually, genesis refers to the creation and generation. So whenever you have like something like, yeah, well, sometimes it's like poiesis, but sometimes often it's genesis. So yeah, genesis is like thinking like it's creating something or generating. So genesis is generating something. So if you put these three root words together, what are you doing? You're generating new glucose. And that's what gluconeogenesis is. It's creating new molecules of glucose. So it's not like breaking down glycogen. It's actually synthesizing and making an entire new structure of glucose. Now the liver is very important. I mean, it's important period. But one of the many reasons it's important is that it is the main site of gluconeogenesis. And gluconeogenesis, even though this is where people get confused, is often that they think it's the same as glycolysis. Not the same thing. It's not the same thing as glycogenolysis. It has a lot of layers in common, but it's not the same process. But gluconeogenesis refers to making new glucose. Now, I don't expect you to know the organic chemical structure of glucose, but I have to talk about the structure of glucose a bit. So yeah, we have this chemical structure, and remember whenever you see those organic chemistry structures, whenever it's not explicitly made, I don't explicitly point out those where the atom is at one of these angles, it's a carbon atom. So we do see, okay, we see oxygen, and these three balls here are hydrogen. But you actually have all these carbon whenever there's like not a labeled atom. So what we see with glucose is that there are 6 carbon atoms, and this number is important, especially when we talk about metabolism later on this semester. But what I want you to focus on at this point is that yes, glucose is a carbon-based structure. But what does how does it involve? What is the role of this structure with gluconeogenesis? Well, again, we're creating new glucose, and carbon is an important atom in the structure of glucose. So what we have are, again, don't worry about memorizing these structures because this is not an organic chemistry class, but what I want you to appreciate is that these structures have carbon in them. So these are all metabolites naturally found in your body, and they're often byproducts of nutrient metabolism and breaking those nutrients down as well. It's like amino acids, glycerol, fatty acids, ketones, they are all that have carbon in their structure. So what your liver can do is take the carbon from all these breakdown metabolites of nutrients and shuffle around the carbons so that you make new glucose structures. So this is a cool thing about your liver. Like say your body needs sugar, but you have all this protein and excess protein and fat in your body. Your body it's not a very efficient process, but it is possible for them to scavenge all the carbon from nutrients and stores throughout your body and make new glucose via this process. And then this glucose, why would you want to do gluconeogenesis? Say your bloodstream doesn't have enough sugar. If your cortisol is telling the liver to increase its rate of gluconeogenesis, it's going to make new glucose molecules to feed your blood so they can feed the rest of your cells. So gluconeogenesis, what do I want you to know? Do I want you to know every single step, every single enzyme, every single metabolite? No, I don't think that's reasonable for this class. Maybe in a biochemistry class, they might ask you about that. But for this one, I want to appreciate that we have carbon in many of the molecules in our body, including the nutrients we absorb. But your liver is able to take these carbon-based molecules, shuffle them around, and then create new glucose molecules. So this is why it's very important to know that glucose has that carbon skeleton. This is a major underlying principle behind gluconeogenesis. Yeah, so it's also involved, it's not necessarily, or breaking down fat, like lipolysis is one way you can generate the carbon needed for gluconeogenesis, but it's not always dependent on lipolysis. Usually they go hand in hand, but yeah, it is related, but it's not the same thing, yeah, for lipolysis and gluconeogenesis. They're all related though. Alright, so let's talk about blood glucose, sometimes commonly just called blood sugar. And when they say blood sugar, they're referring to blood glucose because it's the most common and easily converted into energy in your body, so usually that's what they're referring to. Okay, so this is what's the normal range of blood glucose. So it refers to the concentration of blood glucose in your blood. So again, concentration is how much of something you have in a constant volume of something. So is there a lot of it per volume, or is it very dilute and not too much of it per volume? Now, the normal range and concentration of blood glucose in your body is 70 to 110 mg per deciliter, so milligrams per deciliter. Now, look at this person testing their blood. What they're doing is, hopefully with the second one, but what they're doing is testing their blood and seeing what's the concentration of sugar. Looks like 254, so this person is way above the normal fasting range. Now, it's not the same as liter, but if you're going to nursing and medicine, especially nursing, you gotta know what a deciliter is, because this mg per deciliter appears in so many lab values. Deciliter is not the same as a liter, and it's not 10 liters, it's actually 0.1 liter, so 1/10 of a liter. So that's what a deciliter is. And what is fasting? So I think a lot of you might know, like, intermittent fasting is super popular these days, or is it so popular? I don't know, at least within the past few years. But fasting, what is it? Like, people think, okay, you just don't eat. So this is pretty much what you do. You don't eat or drink, and depending on the institutions, most of them say at least for 8 hours, some say 12 hours. Depends on the hospital, depends on the country, depends on the institution. Like for drinking, most of them accept water, because why? Well, water, if you drink too much water, yeah, that will affect the concentrations of things in your body, but your body also regulates this amount of water, so assuming your kidneys are okay, water is okay. Tea and coffee, this is actually, to this day, is kind of controversial, because it's like, basically they don't want you consuming additional calories or anything that can introduce or lower your blood sugar. So this is why they tell you not to eat or drink, because if you eat something sugary, yeah, what's going to happen to your blood sugar? It's going to go up. But whether tea or coffee could affect these levels, like, even though they might not have calories or minimal calories themselves, depending on the study, depending on the institution, they say it might affect your blood glucose. Like, maybe not, it doesn't have calories itself, but is it going to affect your energy stores, and is it going to cause your body to break down or release more sugar into your body? So that's the point of contention, but water is generally okay. So this is homeostasis, and this is the lab values, and I believe, unless they changed it, snuck it into me, like, I think this is consistent between our books. So 70 to 110 mg per deciliter when it's fasting, so not just like any old time. After you haven't eaten for that 8 to 12 hours, depending on the hospital, depending on the lab, this should be the normal range if you don't have diabetes mellitus. Now, blood glucose levels fluctuate during the day, and this is why it's important to do that fasting, because why? Well, look at this right here. What we see is that if you have a random glucose test instead of a fasting, that means you just to get blood work done, and you didn't really do that fasting period. So it could be after any meal. And look at this chart right here. So what we see is this person's blood glucose in concentration units of mg per deciliter. And notice that sometimes it's as low as almost to 100, and sometimes it spikes almost like around 270. So there's a big swingy range right here. So if you measured randomly throughout the day, you might get something as low as like just above 100, or you might get something as high as 270. So this is why it's important to have that fasting, because it kind of controls this. Now, what's causing all these spikes? Well, notice that where do you see these spikes, and how are they timed? Well, just before each of these spikes, you see these little dips right here. And oh, there's something B, L, and D. Maybe that stands for breakfast, lunch, and dinner, and S might be a snack. So what we see is that shortly after a meal, your blood sugar spikes. And especially if you have like a carb-rich or sugar-rich meal, yeah, definitely that's going to go into your intestines, and your intestines are going to carry that sugar into your bloodstream. Yep, so this is the thing, that blood glucose increases around 22 hours after a meal. Can't fasting for long enough cause glycolysis or gluconeogenesis and a rise in blood glucose. Yeah, so that's the aim of the fasting, is that it's trying to minimize this, but also, like, yeah, your body is going to try to keep your bloodstream fed and maintain that normal level of blood glucose. So that will go on in the background. Like, say okay, you stop eating and your blood glucose drops lower. Well, your hormones will be released, which we're going to cover in this lecture, that will try to bring your blood glucose up to normal. Or say there's too much of this going on and there's like a high rate of gluconeogenesis, maybe your body's going to store this. So hopefully during fasting you have normal regulation of hormones that regulate blood sugar, and it's trying to kind of basically reduce this kind of like swinginess with what you see with just eating whenever, and try to see like, okay, what happens to your blood sugar levels when your hormones and your homeostatic mechanisms take over. Alright, so what are normal random glucose levels? Well, the thing is that, look at this, like take any of these random points and you'll see that there's a huge range. So the CDC and ADA say anything below 200 mg per deciliter, and then Medscape says 140, and then UCSF says less than 125, and then the Endocrine Society says we should actually put it down lower to 100 mg per deciliter. Now, this is showing, I'm not asking you to know this. Why? Because you see that there's a huge range and nobody agrees about this there's no consensus with random glucose levels. I mean, I think I see more people talk about the 200, but it's just showing you that there's differing opinions. Like this is a paper, I think in 2015, and they said like, okay, we're missing undetected diabetes because our cutoffs are too high. But, so this is why we do the fasting blood glucose. We want to measure average blood glucose, but the thing is that, what if somebody, say they did a workout, and this is why they tell you not to do like a workout just before a blood test, because why? Well, that's also going to mobilize your energy reserves, and that can affect your levels of blood sugar as well. So because blood sugar during our day, and it's very easy to cause spikes in it, is there a more reliable way to measure blood glucose? And there is something else so it's not just no, you're not going to take just one fasting blood glucose test and it's high, and they say, oh, you have diabetes mellitus. They're not going to do that. They'll probably retest you to make sure, okay, it's not a one-off, or say something happened and something is an anomaly. But there's another test they can do. And this is, you may have heard this, and I swear, like if you watch TV or even YouTube that's not free from ads these days, you might have heard the term A1c. They're saying, oh, my A1c levels improved, my A1c levels dropped. But the thing is that red blood cells, and these are the cells that contain this protein called hemoglobin, which we're going to cover much more in detail soon in the semester. But hemoglobin is the main oxygen transporting molecule in your body. Now, what the hemoglobin is a protein, but the thing is that glucose, it can chemically bond to the amino acid structures and residues on hemoglobin. So the thing is that red blood cells live about 3 to 4 months in your body. I think the OpenStax book says 120 days, which is around 4 months. But the thing is that, okay, red blood cells, during their lifetime, they're going to be their hemoglobin is going to be exposed to sugars, and glucose can easily bind to the chemical structure of hemoglobin. So normal A1c is less than 5.7%. So when the red blood cell is made, it's not going to have sugar attached to its hemoglobin. But as it lives out its life, around 3 to 4 months, then it's going to encounter the sugar that's floating around in your bloodstream. And the more sugar there is, the more chances it will attach to the hemoglobin structure. And here's a little visual example. So again, here are red blood cells, and again, red blood cells are full of this protein called hemoglobin. And say we have a patient with normal fasting glucose concentrations in their body. So again, during the life cycle of this red blood cell, it's going to encounter glucose molecules in the blood. It's always there. And then during its lifetime, it's going to have this glucose molecules chemically binding to hemoglobin. Now, what if you have a patient with chronic, that means chronic meaning like it's not just you have one little spike of hyperglycemia, which is above normal ranges of the blood glucose. So say this person has almost constant levels of high blood glucose. Well, with high blood glucose in their blood, they're going to have a very high concentration of glucose compared to the patient with normal glucose levels. So with more glucose flowing around the bloodstream, that means more glucose is going to attach to hemoglobin during this red blood cell's lifetime. So what we see is that if you measure the amount of glucose attached to hemoglobin from these red blood cells, you'll see that this patient with chronic hyperglycemia has more glucose attached to hemoglobin. So that's what we were talking about when we're referring to hemoglobin A1c. So the interesting thing about it's kind of like a snapshot of the last 3 months of your blood sugar content. So this is why often fasting glucose tests often go hand in hand with this HbA1c testing. Like someone can be like hyperglycemic or just eating crazy amounts of sugar for like the first maybe until a week before their A1c test. Like just fasting for 1 day is not going to lower your HbA1c. This is again over the magnitude of at least the past 3-4 months and your red blood cells being exposed to sugar over those 3-4 months. Okay, so the thing is that your body so blood glucose is always, the normal fasting range is that 70 to 110, but your body can use its reserves to add to that blood glucose pool. So we have glycogenolysis, and if you remember your root words, lysis refers to something that's breaking down, and glycogen is that big branched macromolecule where you have all these glucose molecules daisy chained and they're branching off into this structure that looks like a little tree of glucose. So when you break down glycogen, you're taking that glycogen and breaking down into individual glucose molecules. Gluconeogenesis is what we just covered. You're going to again take random, or not random nutrients, but you're going to take carbon-based molecules in your body, and especially the liver is going to shuffle around and create new glucose molecules. So the other thing is that your body is not only just creating its own glucose, but your body can easily get glucose from external sources. And how? You just cow, right? You just eat. So how do you do that? Well, we can just, easy like that. But this is the gross anatomy of the pancreas. So we get to this when we talk about the digestive system, but this is talking about the exocrine system and how it's actually able to secrete pancreatic juices into the digestive system. Now, what we don't see, so the pancreas is not just endocrine, nor is it just exocrine. This actually involves both processes. So exocrine involves secreting onto some cell surface and like exocrine secretions like sweat glands, but actually this part right here, secreting into these ducts, there is an inner surface in your body, so even though it's not exposed to as exposed as your skin is to the environment, it still is a surface inside your body which exocrine secretions can occur. So that would be the exocrine function of the pancreas, but you also have the endocrine functions, and that's having those molecules that enter the bloodstream and stays inside your body until it leaves via the urinary system or if it's broken down. But endocrine, so my main point is that the pancreas is able to do both exocrine secretions into your gastrointestinal tract, but it also can secrete chemicals into your bloodstream. Now, this is the pancreas, it's showing more of the exocrine anatomy and ducts in this picture. And in the Martini, they don't have a picture like this, and I don't think the OpenStax really has one either. But there's a lot of there are blood vessels that carry blood to and from the pancreas. So this is more of the hormonal regulation where, again, if the pancreas sends the cells that produce hormones in the pancreas to make something, it has to be carried into the bloodstream. And that's what we're seeing with this vasculature and blood vessels right here. Now, again, pancreas is not just exclusively exocrine or endocrine, it has functions in both. Now, the exocrine involves those digestive enzymes we'll talk about later in the semester, but endocrine refers to the production of hormones into the bloodstream. So the pancreas produces hormones. What are the hormones? Well, they involve these little things called pancreatic islets. So again, we have, or if you're here on the island, hopefully you are, that we are on an island and we're surrounded by water, right? So we have a little small island right here, and this is like a little islet, and small island surrounded by this body of water. Well, in pancreatic islets, we have these small group and clusters of cells, and these are endocrine cells. And depending on the type of cells found in these islets, they produce different types of pancreatic hormones. So this is all called pancreatic islets, or you might commonly hear the term islet of Langerhans. And I know there's always, it comes and goes in ways where we're trying to get rid of like things named after people, but it always comes back, and especially if you're working with more seasoned healthcare professionals, they probably know these, so it's worth knowing that islets of Langerhans is referring to these pancreatic islets. And then you have these pancreatic acini, and these are clusters of exocrine cells. So just like this little islet is surrounded by ocean, these pancreatic islets are surrounded by exocrine cells. So the islets are endocrine cells, the surroundings are exocrine cells. Alright, so there are 4 main types of pancreatic islet cells. So the first 2 I definitely want you to know. The other 2, it's not that they're not important, but they're a little more complicated, and maybe your advanced classes might talk about them more. So alpha cells, and this is the Greek lowercase letter alpha, they produce a hormone called glucagon. And beta cells, they might produce a very familiar hormone called insulin. So, and then there's also these cells, and if you alpha, and if you know your Greek alphabet, alpha, beta, gamma. So gamma cells, but more commonly now called PP cells. PP stands for pancreatic polypeptide. And in other textbooks, they also call them F cells. But yeah, I've rarely heard them call it gamma cells. Maybe when they were first discovered, they were called gamma cells. But it seems like the hormone they produce is more like that kind of took over their name. And the cool thing about this pancreatic polypeptide is that it decreases hunger and decreases like appetite as well. Now, delta cells, they produce something called somatostatin, and another name for somatostatin is GHIH, or growth hormone inhibiting hormone. And it's not just inhibiting growth hormone, but delta cells also, somatostatin, also inhibits the production of glucagon and insulin. So the last 2 are a little tricky, and I don't know if we have time to really get to that this semester. But alpha cells and beta cells, you gotta know this if you're talking about blood glucose, the blood glucose regulation and homeostasis. So insulin, again, look at this structure. What do we see? A ribbon of amino acids all daisy-chained together. Therefore, it's a polypeptide. Therefore, it is a peptide hormone, a protein hormone. Now, these are released by pancreatic beta cells. So again, it's not alpha cells that produce insulin, it's beta cells. And beta cells, and what's the major effect of insulin on glucose? It basically allows glucose to be taken in from the bloodstream and into cells. So what's the overall effect? Well, if you're moving glucose from your bloodstream into your cells, that's removing it from the bloodstream. So if glucose keeps disappearing from your bloodstream and entering your cells, that's going to lower your blood glucose. But it's also going to feed your cells as well. So again, what happens when this glucose enters your cells? Well, glucose is very easily metabolized by your cells and converted into energy, especially that very important molecule ATP, which is used for storing energy and also providing energy for chemical reactions. And it's also going to increase glycogen formation. So now that you have with insulin, you're bringing all this glucose into cells. What is it going to do with, if it has, okay, I love all these sugary snacks, but I might have a little too much. So it might put it aside for later when it actually needs it again. So glycogen is a very important storage form of glucose. And it's also going to increase the absorption not of just glucose, but also other amino acids, and also increase protein synthesis as well. And it's also going to increase triglyceride formation, so that's a form, one of the major lipids in your body, or forms of lipids in your body. So what we see is that it's kind of building up glycogen, it's building up proteins and synthesis, and it's also building up triglycerides and fat. So why? Because you're bringing all these nutrients into your cells, and your cells are going to store it for later, or make good use of those nutrients and build itself up. So even though like some endocrinologists don't like the term anabolic and catabolic, insulin is kind of an anabolic hormone. Because why? It's building up molecules, it's taking smaller molecules and building bigger structures. So I think there are some people who, like bodybuilders that like using, or powerlifters that might use insulin. Why? Well, if you're trying to be very lean, yeah, I mean, you might build a lot of protein, and you might feed your muscles with glycogen, but you're also going to increase your adipose production of triglycerides. So this is why insulin may be anabolic, but you're also building a lot of adipose as well. Not that I'm endorsing it, but I'm just letting you know that insulin is very important for feeding and building things up. Disclaimer there. Okay, so insulin, what is the basic mechanism and how does it affect the cells? So what happens, this is what I want you to know for this class. So we have insulin, our peptide hormone, and again, for a cell to respond to a hormone, it needs some sort of receptor. So insulin is a peptide hormone, and not coincidentally, it's going to have its receptors on the outside. Why? Because insulin is water-soluble, so you want those receptors for insulin facing the water-soluble surroundings of a cell. Now, when insulin binds to its receptor, it's going to cause these intracellular changes, and this is called signal transduction. Now, I'm showing you as a cartoon, but I'm just letting you know that these enzymatic and chemical changes occur inside the cell. Eventually, this triggers these vesicles that are inside the cells to move to the surface of the cells. So these vesicles, these little bubbles in membrane, they have embedded in them something called glucose transporters. So these are going to transport glucose. But if they're not facing the outside of the cells, they can't really transport anything. So if you move these glucose transporters to the surface, so the insulin binding to the insulin receptor is going to trigger the movement of these glucose transporters to the surface of the cell, and now they're appearing on the surface of the cell. So when these glucose transporters appear on the surface of the cell, they have all this glucose floating around the vat in your extracellular fluid and blood. Now these glucose transporters through diffusion can move, or glucose is going to move into the cell via diffusion, and these glucose transporters are actually facilitate diffusion. And these glucose transporters are important in this aspect. And this is what I want you to know. So insulin binding to insulin receptor, it causes glucose transporters to appear on the cell surface. When glucose transporters appear on the cell surface, it can move glucose into the cell. So this is what I want you to know. And this would be what the med students have to know. So very similar things, we have insulin binding to its receptors. And we have the appearance of these glucose transporters. That's what GLUT4 is. And GLUT4 is going to bring glucose in. Now the rest of the stuff, I don't expect you to know this because this is more of the molecular stuff. So this is the med school level stuff. And if you're at the PhD level and you have to if you're studying these pathways, this is what we study at the PhD and doctoral level. So if you're want to have a degree and doctorate in cell molecular biology, this is what you're up against if you're studying the glucose pathway. Again, I don't want you to know this crazy pathway right here. I mean, I know I had to study it, but it's like, I don't expect that at this level. If you're studying this, you're going way beyond the scope of this class. That first cartoon is what I want you to know. So I was giving you the easy version, that first slide. Alright, so let's put this with this mechanism of insulin and glucose transport together with blood glucose. So here we have the bloodstream, and we have all this glucose floating around, and we have all these cells. And then we're also going to have our beta cells producing insulin. And insulin, if we have all these insulin receptors on these cells, what are these cells going to do? If insulin binds to these insulin receptors on these cells, this is going to cause glucose transporters to appear on the surface of these cells. And if these glucose transporters appear, where is the glucose going to go? It's going to go inside these cells. Now, what did you notice before and after all of this? Notice that the concentration of glucose in the bloodstream dropped. So it's not like the glucose just evaporated into nowhere when you add insulin, but by moving glucose from the bloodstream into cells, you're removing glucose from your blood. So this is why insulin lowers blood sugar. It's because you're just moving it from the blood and allowing cells to take glucose from the blood, thus lowering blood sugar concentrations. Now, glucagon. So what do we see here? We see and if you don't know what these three-letter words are, these are all amino acids. So glucagon is another peptide hormone. And here's the 3D structure. So it kind of forms this like this helix right here. So this is an alpha helix, but actually that's more for like molecular and cell biology. Alright, so it is a peptide hormone because it's made from all these amino acids being linked together again. And then what is made by these alpha cells? So again, insulin is made by beta cells. Insulin is a peptide made by beta cells. Glucagon is a peptide made by alpha cells.
Now, what it does is free energy reserves into blood. And what do I mean by that? Well, it's going to basically take the parts of the different ways you're able to store glucose and nutrients in your body, and now it's going to break them down and release those nutrients into your blood. So in skeletal muscle and in the liver, instead of doing that gluconeogenesis or glycogenesis, it's going to actually stimulate the catabolism. It's going to cause glycogen to break down into glucose. And then adipose, now we have so adipose is going to cause your adipose tissue, if you have glucagon, it's going to cause it to break down triglycerides into fatty acids. So triglyceride is the storage form of these fatty acids, and fatty acids, they're able to be transported in your blood and also being broken down into metabolites or nutrient, or actually be broken. Well, I won't get too far ahead in this part because we talk about metabolism, but you're actually able to break down fatty acids into energy. That's what we're thinking here. Alright, so in liver, what does it do? It stimulates the production of glucose in the liver, so it also stimulates gluconeogenesis. So just actually, you might be thinking, wait, we talked about breaking down glycogen and freeing up glucose and dumping it into your body. And we also thought, or bloodstream, and we also thought of okay, wait, gluconeogenesis, that was the liver, but it was a different hormone that was stimulating it. So this is an interesting thing. Glucagon and cortisol, so cortisol stimulates gluconeogenesis, but so does glucagon. So cortisol is going to stimulate gluconeogenesis, and you're going to create new glucose molecules, which end up in your bloodstream. Now, what is glucagon going to do to adipose tissue? It's going to cause it to break down triglycerides into fatty acids and glycerol. And glycerol is a carbon-based molecule that can actually be used for gluconeogenesis. So we have the production of even more sugar via gluconeogenesis. And then glucagon is also going to break down proteins, and here we have skeletal muscle. So it's actually able to break down proteins into amino acids. Amino acids can also be used in gluconeogenesis. So if you have amino acids going to gluconeogenesis, now you have even more blood sugar. What do we see here? With glycolysis, and actually it should include glycogen or glycolysis as well, or not glycolysis. I should also use the glycogenolysis and see the breakdown of glycogen into glucose. That's going to add even more sugar to the bloodstream. So what we see is that glucagon and cortisol, they do the opposite of insulin. Instead of building things up, they're breaking things down and creating more glucose molecules. And what we see is that before and after, we're increasing the amount of sugar and glucose in the bloodstream. So glucagon and cortisol are going to increase the levels of blood sugar in your body. And glucagon, so this is why I definitely want you to know glucagon and insulin, because not all hormones have opposing action. There are many hormones, and classic examples insulin and glucagon, that have opposing actions on each other. We will cover a few more before we wrap up the endocrine system, but glucagon and insulin are a classic pair. One increases the blood glucose levels, the other decreases blood glucose levels. But I also want you to appreciate how sometimes hormones don't just oppose each other. In terms of glucagon and cortisol, we have two different hormones, but they're able to work together. Alright, so let's wrap up and talk about something called Glycemic Index. And what does it do? Well, we have this blood glucose levels, and again, blood glucose levels are going to change in response to eating something, right? And diabetes mellitus, we will get to that, so it is coming up. So yeah, I'm glad you're asking about that, but we'll cover that, don't worry, just hang in there. Alright, so what we see is that we have a spike, but with a low glycemic index, you notice that spike doesn't have as much amplitude. So the maximum level you reach isn't as high as it is here with this high glycemic index. But it's not just about the maximum blood sugar it reaches, but also the timing as well. So we see that in a shorter time, around maybe half an hour, we see this huge spike. But compared to that same time, notice that this peak takes a little longer in this low glycemic index food. So this is why you might hear slow release of carbohydrates and sugar compared to a high glycemic index where it's almost immediately absorbed into your bloodstream. So the glycemic index rates food on how they affect blood glucose, and they rate it on a scale, numerical scale,
And pure sugars, glucose is supposed to be 100. Now, lower glycemic index, so if you pure glucose is easily absorbed by your body, so if it's going to cause a very sudden and short spike right here, and basically it's going to compare this and see, okay, what's the glycemic, how does it compare to pure glucose? But if you have a lower glycemic index, this, instead of having this rapid and this rapid spike in blood glucose, it's going to release and add glucose to your bloodstream slower over time. And again, higher the glycemic index, that means it's closer to 100. This is going to cause a sharper increase in blood glucose. Now, again, remember why? Because again, we have our blood glucose always spikes in due to whatever we eat throughout the day, and when we eat, but also what types of food we eat. And again, so just so blood glucose by itself, day to day, or even on the order of minutes, can change. Now, again, so what we have, eat, blood glucose goes up, eat, blood glucose goes up. So this is how they're able to measure blood. And how do they do that? They actually measure what we call the area under the curve. And this is what they're showing the shaded part. How do you measure the area under the curve? Well, if you're taking a calculus class, that might sound familiar. What are you doing when you take an integral? You're actually measuring the area under the curve. So you're like, oh, when do I use calculus? Well, glycemic index is an example of that. Alright, so let's go back to top hat. And let's ask a little question about, hopefully it works this time. So which of these foods has the highest glycemic index? And I controlled for weight, so say you have 100 grams of white rice, 100 grams of brown rice, or 100 grams of baked potato. Which do you think is going to spike your blood sugar the most? Give you the highest spike in the fastest time. Okay, let's see what the responses are. So most of you said white rice would have the highest glycemic index. Second place, a potato. Last, brown rice. Well, what's the answer? Well, what answer is there? It depends on who you ask. I know it's not going to be very satisfying. And what we see is, so which of these has the highest glycemic index? White rice, brown rice, or a baked potato? Well, it depends on the chart you're using. Let's look at those things over here. Now what we have is like glycemic index vary depending on who's doing the measuring and the study. So let's look at these things. What we looked at white rice. In this chart it says 72, and it says brown rice is at 66, and a potato is at 80. Over here, brown rice and white rice. Again, white rice still has a higher glycemic index in this chart compared to brown rice. And then it's this chart. It's interesting because it says that brown rice has a higher glycemic index than white rice. So we're having this kind of flip-flopping happening over here. Who do we trust? And this is what I'm trying to say here is like, I got all of these just by doing a simple Google search for glycemic index charts, and you'll find out these values often vary between these. So yeah, we have our potato here, and potatoes tend to be, yeah, it's pretty consistent among these charts. But, oh yeah, over here, look. Over here with this left chart right here, it's saying that potatoes are almost like pure glucose. Whereas over here it's saying like, okay, it's like 85 glycemic index of 85. Now, this is my main point, is like, don't just rely on one source on the internet. And what's a better way of doing this? Well, another way you can do this is like, say you have a lot of institutions or studies that disagree. One way is that to take a bunch of different studies and do what we call a meta-analysis. So this study right here that was published in Diabetes Care, sponsored by the American Diabetes Association, is that it took a bunch of these articles, and these are peer-reviewed articles, so someone had to look over the methodology, and they took over almost a few decades. But they took over multiple years, and not only did they just use the values, they did some vetting as well. So they're like, okay, are we going to talk about the glycemic index in people who already have diabetes or people who are not diabetic? So this is what they looked at. Or sometimes they also eliminated studies that had too few subjects. Like that means like they got glycemic index from like less than 4 people. If you take statistics or biostatistics later on, you'll know that statistics are more reliable and less prone to errors due to random variability and chance the more people you have in the study.
So yeah, having less than 5 people or fewer than 5 people is like, or 5 people or fewer, that's like not a very well done study. Or wide variability. Say the glycemic index is like the range of them of these measurements was too wide, so they have the standard error to the mean. So that I won't. This is a statistics class, but it's just saying like compared to having results that are very close to each other, these studies with this high standard error to the mean, they had like results all over the place. So they weeded out and that's another important part of doing these analyses. You have to kind of weed out low quality studies or studies that didn't use enough people or something was inherently flawed with their methods. But you also don't want to compare people who already have diabetes mellitus with people who don't have diabetes mellitus, because people with diabetes mellitus, as we'll cover in future lectures, have a different response to insulin and blood glucose. So what they did is like, okay, they're looking at various different vet foods, and they're going to take these glycemic index, and notice that they have a plus or minus. So it's saying that in terms of this range, they're using statistics to generate a range where they think the actual value is. So in this one, what we see is brown rice has a slightly better glycemic index than white rice. So this is more a better answer than just going over some random chart you found on the internet, because why? Now where they're looking at more studies and they're trying to use statistics to come up with a better value that uses more people rather than just going on one small sample of like 5 or fewer people. But this is also very interesting too. Yeah, oh yeah, potatoes, they have generally a higher glycemic index, and notice that boiled potatoes have a lower glycemic index than having mashed potatoes. Well, why is that? Well, your digestive system, when we talk about it, it has to break down things and process things and use enzymes to digest things so it can enter your cells. So mashed potatoes already does a lot of the work for you. Your body doesn't have to chew it up or break down the starchy tuber. It's already kind of mashed up, and all you have now your enzymes can digest the potato more efficiently. I like this part too, taro, right? So kalo, poi, right? So this is a great thing. My grandma has like type 1 diabetes, and this is one way she was able to manage her blood sugar. Like she can't have candy because that's going to cause her blood sugar to spike, and then she has to like reach for the insulin. But she switched from, even though it was tough for her at first, she loves her rice, but she switched to poi, and she eats it as her main starch, and her blood sugars have stabilized because why? Well, we see that boiled taro has lower glycemic index, so it releases the blood sugar, so her blood sugar wasn't spiking as much. Now, does that mean like, okay, I'm going to go switch? I have to know someone who has diabetes. I'm going to tell them to switch to taro. Don't do that without medical advice, but it's just something she did, and she saw that it worked for her. But there is some evidence. We see that taro has or kalo has a low glycemic index. All right. So again, this is about blood glucose, and this is the reference value we'll use. And again, I point this out because there are different institutions that use different units, or sometimes the range might differ. This is the range we're using for our class for PHYL 1 for 2, and we're not talking about random blood glucose tests because again, if you eat something, your blood glucose can go up. So fasting range is 70 to 110 mg per deciliter. The time of fasting, I'm not going to test you on that, but you should know what fasting is. And then hypoglycemia. So let's talk about what happens when you have too low blood sugar. So again, high blood sugar can cause a lot of problems, but if you have your blood sugar dips too low, you'll feel tired, you feel pale, and if you've had shakiness, like and anxiety, sweating, and hunger, so yeah, when you have hypoglycemia, sometimes people think it's a panic attack, but they might test their blood sugar and be like, oh my god, you have a low blood sugar, blood sugar, and this is what's causing all your symptoms. So what's an easy way to counter hypoglycemia? Well, assuming they're not diabetic, you can just eat something sugary. So that's why people who might be prone to hypoglycemia, they might carry a sugary snack with them. Hyperglycemia, what happens if your blood sugar is too high? Well, you're going to get something called people with uncontrolled diabetes mellitus. They often have hyperglycemia. We'll get to why in a future lecture.
So one thing is that they have polyuria. So poly meaning many, and urea sounding like urine, right? So this means many urine and increased urination. So again, your kidneys are important in taking your blood contents and adjusting the chemistry of your blood. But what if you have a lot of sugar in there? Well, through principles of osmosis, if you have a high concentration of solutes such as sugar, water is attracted to sugar, right? So if you have a lot of sugar in your blood is going to hold on to a larger volume of water. When this sugary blood enters your kidneys, you're also going to bring that increased water volume to your kidneys. If you have an increased water volume in your kidneys, how is your kidneys going to maintain fluid balance in your body? Well, it's going to produce more urine to get rid of that excess water. But a consequence of this is that usually your kidneys, they're functioning normally and you have normal levels of blood sugar. They're able to reabsorb sugar from your blood and recycle it back into your bloodstream. Now, what we saw there is that if you have too much sugar in your blood, sometimes it can end up in your urine. And that is something we call glycosuria, or abnormally high glucose in your urine. You should be near zero, but if you have a high presence of a lot of sugar in your urine, that's what we call glycosuria. And remember that you're having a lot of water to go out with the sugar and all of this urine. So this is why another symptom of hyperglycemia is polydipsia, because with all this water entering your blood, all this water being filtered by kidneys and dumped into urine, well, you're going to lose a lot of water from your body. Therefore, you're going to feel thirsty. So this is polydipsia. And you have a lot of blood vessels in your eyes. This is why blurred vision is also affected by hyperglycemia. And you also have fatigue as well. So again, fatigue is basically saying if you have blood sugar that's too high, you'll feel tired. But if you have blood sugar that's too low, you'll also feel tired. But it depends on what other symptoms you have. So you can't just make a diagnosis based on just what symptom. You should do it like many conditions in your body, they have a certain set of things that go along with them. Or actually, I'm not going to go into differential diagnosis, but yeah, just showing you that this is why it's important to have a normal level of blood sugar.